Hello everyone, welcome back to my single cell RNA sequencing data analysis video tutorials. Today I'm going to make a second fans requested video. So if you see from my comments, one of you asked me to help to change ensemble ID in your Seurat object into gene symbols. Because you are going to create a cell chat object from a Seurat object, today I'm going to make a video to show you how to change the gene ID in Seurat object. But it is also possible to change the gene ID in your cell chat object. So let me go back to R and show you how to do this. So first we need to load the libraries Seurat tidyverse, and also we load the human ensemble database because in my second part I'm going to show you how to convert gene IDs into gene symbols. So I'm going to use a long dataset. I downloaded this dataset already, you can see in my computer. I have compressed the files, barcodes, features, and matrix data, and also I unzipped the feature files here. It will depend on what kind of data you have. If you have the feature files, and normally the feature files contains the gene ID and also the gene symbols. So if your feature file has both gene ID and gene symbol, it will be easy to change gene ID into gene symbols. So let me show you how to change gene ID to gene symbols if you have a feature file contain both gene ID and gene symbols. So first let's use the 10 times function to load the data. And also we can load the feature files in. So you can see we have a large DGC matrix data named as long. If we have a look at the DGC matrix data in the dim names 1, you can see when we use the 10 times read function to load the data, the row names are the gene names. Because your problem for the data analysis is that you have the gene ID name here, so we can change the row names to gene ID so we can get the information from the feature files. You can see in the feature file, the first column V1 has the gene ID, and the second column V2 has the gene symbols. So we can use the column 1 information, gene IDs, to change the gene symbols in the data set. So let's have a look at the long data again. You can see we have 33,694 genes, and in the feature files we have the same number of genes here. And in the dim name for the row names, at the moment they are gene names, so we can change the gene names into gene IDs because we know the V1 column in the feature files are gene IDs. So let's change it, and we can have a look at the long data again. If you look at the dim names now, you can see row names are gene IDs, they are not gene names anymore. So we prepare a dataset that the row names are gene IDs. So now we can perform a standard Seurat analysis. We can create a Seurat object, keep genes that expressed at least in 3 cells, and keep cells have at least 200 gene expression. So we created the Seurat object. If you look at the Seurat object now, we only have 12,355 genes in the dataset. So now we can go through the standard Seurat workflow analysis. First, we normalize the data, then find the variable features, then we scale the data. After that, we run PCA. You can see now we have the gene IDs, not gene names. So we can find the neighbors, then find the clusters. You can see we have 7 clusters here. Finally, we run UMap. Then we can use the dim plot to look at the cell clusters. So you can see we have 7 cell clusters here. So after analysis, we can check the dataset again. For the cRNA in the count, the dim names, the row names are Gene IDs. So now we have analyzed the Seurat object. The row names are Gene IDs, not gene names. So I will use this dataset as the example to show you how to change the gene IDs into gene names. Just now I showed you we saved the gene IDs in the data slot, cRNA counts and dim names 1. So we can get the gene IDs from the Seurat object. You can see we have 12,355 gene IDs. So I showed you in the feature files, we have 33,694 gene IDs, and also this file contains corresponding gene names. So we can use the gene name information from the feature files to replace the gene IDs in our Seurat object. So you can see at the moment the feature files, the row names are in numbers. So we can set the row names as gene IDs for the feature files. So we know the V1 column has the gene ID names. So we can use the V1 column information to set the row names as gene IDs for the feature files. If you have a look at the feature files now, you can see the row names are gene IDs. So because the feature file has 33,694 genes, but the Seurat object only has 12,355 genes. So we can use the gene ID information from the Seurat object and subset the feature files. So let's do this. You can see now the feature files only has 12,355 genes, and it has the same number as the gene ID. So now the feature file has the same number genes as the gene ID we have in the Seurat object. Let's add the gene ID from the Seurat object into the feature files as the third column to see actually if they match each other or not. We can have a look at the feature file again. You can see now we have the third column. You can see the gene ID in the third column match to the first column from the feature files. That means we created a correct file contains the gene name for all the gene IDs in our Seurat object. So the gene name are in the V2 column in the feature files. So we can use the gene information in the V2 column to replace the gene IDs in our Seurat object. So before we replace it, we can have a look at the dim names in the Seurat object again. You can see it's in the cRNA count and the dim names at the moment. They are gene IDs. If we replace it with the gene name, we can have a look at the Seurat object again in the cRNA counts dim names. You can see now we successfully replaced the gene IDs into gene names. So now you can convert the Seurat object into cell chat object with the correct gene names. So I showed you how to use your feature file from your dataset to replace gene IDs into gene symbols. Sometimes in your dataset you don't have a feature file, or you have a feature file only contains the gene ID information. Then we can use the ensemble database to convert gene IDs into gene names. Because the data I have here was generated from human lung tissue, so we need to use the human ensemble database to convert gene IDs to gene symbols. In the first part, we already converted the gene IDs into gene symbols in our Seurat object. Let's have a look at the Seurat object again to confirm it. You can see in the cRNA counts dim names, we have the gene names. So we want to use this dataset to demonstrate how we use the ensemble database to convert gene ID into gene symbols. So we know in our feature files the V1 column are the gene IDs. So we can replace the gene names to gene IDs again. Use the gene ID from the feature V1 column. So let's have a look at the Seurat object to confirm it. cRNA counts dim. So now you can see in our Seurat object the row names are gene IDs. They are not gene names anymore, so we can use the dataset to show second part of our demonstration. So now we can change the gene ID from the dataset as a data frame. If you have a look at the gene ID now, the gene ID column was named as gene ID. So we can use the map IDs function to map our gene IDs into gene samples using human ensemble database. And then we will create a new data frame. We name it as gene name. So you can see we converted the gene IDs to gene names, but 10 of gene IDs were unable to convert into gene names. I compared the different methods. This method is the best one. So let's convert the gene names into a data frame again. Then we can look at the gene names. You can see now in the row names we have the gene ID, and in the gene name column we have the gene names corresponding to each gene ID. So we can add the gene ID as a new column to the gene names data frame to confirm they match to each other. So we can have a look at the gene names again. You can see now we have a new column from the Seurat gene IDs. You can see they match to each other. So now we can use the gene name information from this data frame and replace the gene ID in our Seurat object. Before we do that, we can double check the Seurat Object to make sure at the moment the row names are Gene ID. You can see at the moment the row names are Gene ID. If we replace it with the gene information from the gene name and data frame, now you can see we change the gene IDs into gene names. So in my second part, I showed you how to convert gene IDs into gene names using ensemble database. Because my dataset was generated from human lung tissue, if you have a dataset from mouse tissue, then you need to use the mouse database. So okay, that's it for today's demonstration. I hope this video can help you solve your problem, and good luck for your data analysis. I will be happy to hear from you if you managed to change your gene ID into gene names after watching my video. Thank you and see you next time.